Welcome to the Inspiring Minds. And now let's begin with the next session of the microbiology. As you can see in the screen that earlier in my 3 videos I have told you about the microbiology, the basic concepts. That was the introduction of the microbiology, and then I moved toward the morphology and physiology of the microorganisms. Now we are over the question session. As I told you from the beginning that as microbiology contain 10 to 13% weightage in various examinations, and the first video was the gram staining, and also in second I gave the brief introduction about the gram staining in morphological and physiological properties, and I told you about the capsules, capsid, flagella, pili, etc. in my previous video. Now we are moving toward the question session. So I would like to make it clear that our target when we deal with the questions, if I'm right, then we have two sections. The first one where we are going to deal with the crock questions, and the second one where we will deal with the IFM, USMLE and other examinations. Okay. While solving all these questions with the concepts, one thing keep in mind, that your knowledge is going to be testified, right? So now look at the screen. There the first question. That question have been asked 3 times. If I'm right, then more than 3 times in the crock questions. So just read that question. That is a smear of sputum from the patient with the suspected lobar pneumonia was stained with the use of the following stain and reagent: solution of the genital violet, that is crystal violet, Lugol solution, 96-degree alcohol, and water magenta, and what staining method was applied in this case. And there are 5 options which have been given: gram staining, Gill-Nielsen staining, Romanowski staining, Neisser staining, and Leffler staining. Look at me, friends. When you solve the questions, keep pen in your hand and just follow those techniques which I am telling you. Definitely you will have the confidence, and you will also know that yes, you are doing the right approach. Now again see the question and just highlight those things which I am telling, like smear of the sputum from the patient suspected lobar pneumonia. Just highlight lobar pneumonia. The second one is the genital violet or crystal violet. The third one, alcohol, 96 degree, and the water. Okay, now come to the 5 options: gram staining, Gill-Nielsen staining, Romanowski staining, Neisser and Leffler. Look at me, friends. When I told you about the gram staining, the 6 steps of gram staining in my previous video, so there was a short and compressed description about the gram staining. So here in the 6th step of gram staining, there was the primary stain and the secondary, also known as counter stain, that was naming as saffron stain. What we do there, we just identify the gram positive and gram negative bacteria. Okay, now one step ahead. Actually, microbiology is not only the study about the gram positive and gram negative bacteria. There is a lots of other factors which you also need to be remembered. So on the behalf of gram staining, you can't solve all the questions. You should know the further concepts. In these questions, the smear sputum from the patient, as in the question it has been given, that the first one you talk about the gram staining. The first option was the gram, and the second one was Gill-Nielsen, and third Romanowski, and further. What you should know. See, there are 2 ways to approach the question. The first one, you just ram over the questions and answers, that what are those answers, and if you know the concepts behind them, then definitely there is no probability to be wrong in the questions. Okay, I am giving the basic criteria about solving these kinds of questions. When we approach toward the questions, what is our target? The first one is crock after third year, and the second one is IFM, USMLE approach. In both examination, your knowledge will be checked. In the questions, if you follow the NEET PG questions, or in the crock questions and IFM and USMLE questions, they are asked in 3 factors. Somehow they will ask name, sometimes they will ask about procedure, and sometimes those questions will be based on the results or conclusion. technically if you go over them in the down step, then you will find name based questions asked in crop and procedure based question asked in IFM and USMLE, and similarly NEET ask it in combined way. This all things are my personal observation while seeing the microbiology questions. So let's just I'm giving you one example here. Like in croc questions, they say Zeal-Nielsen method. Okay, in IFM USMLE questions, they will simply say the acid fast staining, positive or negative. In croc question, oftenly you will listen the words, you will see the word Romanovsky method, and in the IFM USMLE questions and other license examinations, there will be known as differential diagnosis. In the croc questions, the term is used Neisser's method, similar in the differential diagnosis. In Loeffler method, and also there are few more examples. What is common in them? There are microorganisms which is common in them, and the results of them, like if you on the basis of result, we just conclude that what are our questions, what is our question asking to us. So what is there, the target is always same, okay, but the conclusion may vary because they contain the different staining process, staining component. Because of that we say that they have different results. Simple, if you know that gram staining, because of that saffron staining and then retaining the saffron color, we declare that is gram negative red color. And when there is crystal violet, the previous one, the primary stain containing, that is a gram positive stain. In this way we just declare that this is a gram positive and gram negative, right? Now the further bacterial properties which I told in my previous video, that is capsid capsule, flagella, villi, and also in extreme condition they form spore, the process of sporulation. Okay, in croc questions, just I give just that that question which I am discussing here, it contains itself 5 topics. Don't worry, I will tell you that in the in my fourth video, then fourth session, and also it will be repeated in the next session when there will be specific property about like bacterial spore, or when will we talk about those particular disease like tuberculosis and Clostridium pyrifrangisis causing diseases, wet gangrene, and there also I will mention the differential diagnosis according to the Kaplan notes. Okay, here I am giving you the basic example from which you can solve the question of the crop. Okay, so let's see the one by one them, so that you can know the next step about this staining process. So in this video I told you about these staining methods which have been used. In next video I am going to tell you just comparative analysis of these 4 options. Trust me guys, there is gram stain, Zeal-Nielsen, Romanovsky, Neisser and Loeffler. They 5 themselves are very important, and also definitely you will face a question if you solve the croc question in every year. Okay, and based on their result, every time in differential diagnosis of the USMLE and IFM is been asking. Okay, so in the next video, hope you will continue this video, and the next video you will just see the comparative analysis, so it is going to grip over your mind. Hope you guys like this video, and let's meet in the 5th video, where I will give you the comparative analysis of these methods of that first question, where the 5 concepts will be repeated, and based on them I will tell you next 50 questions how you can solve. Okay, so now just look at them. In the first what I told you that there are the 10% weightage of microbiology, which you should always remember that it is going to give you the basic knowledge how to solve the question and how to approach to them. And these methods which themselves are very important, let's discuss in the next video. So like, share and subscribe this video, and hope you will like this hope you like these concepts, and in the next that will be really very much interesting for you. Thank you.